Hello students, thank you for tuning into this lecture on Chapter 4, which is on the prokaryotic and the eukaryotic cells. You're going to see that despite their complexity and variety, all living cells can be classified into two groups, and those groups again would be prokaryotic and eukaryotic. Prokaryotic cells are cells that do not have a nucleus. Prokaryotic comes from that Greek word pre, and the pro meaning pre or before, and then the term karyote, kary refers to a kernel, and that kernel is actually referring to the nucleus. So for prokaryotic cells, we are essentially saying before a nucleus, whereas in eukaryotic cells, the term means a true kernel or a true nucleus. The differences between the prokaryotic cells and the eukaryotic cells are plentiful. Because of the formatting, I'm making some arrows so that you know which heading goes to which column. For prokaryotic cells, they just have a simple singular chromosome, and it is not in a membrane. So the cell, if I were to draw it here, you would see that their genetic material is not in a nucleus. We just call that a nucleoid region. Whereas the eukaryotic cells have DNA that is contained within a nucleus. There's a membrane surrounding it. In prokaryotic cells, they do not have histones. Histones are chromosomal proteins that are found in eukaryotes. So in the eukaryotic cells, they will definitely have the histones. Prokaryotic cells do not have membrane-bound organelles, whereas in the eukaryotic cells, you'll find several types of organelles that have specialized functions. The prokaryotic cell has a peptidoglycan cell wall, but in the eukaryotic cells, if they have a cell wall, it is not made of peptidoglycan. Instead, it is of some sort of polysaccharide. And the prokaryotic cells have one cell that will divide into two cells, and this is called binary fission. But in the eukaryotic cells, you also have one cell dividing into two cells, but we call our process mitosis, or the process in eukaryotes. We call it mitosis because it has several stages that are associated with it. So in binary fission, the DNA simply is copied and the cell is split into two, and there are fewer structures and processes than in eukaryotic division, where you'd have the different stages: prophase, metaphase, anaphase, telophase. You have the mitotic spindle developing, so it's way more of an involved process. Prokaryotic cells make up a group of very small unicellular organisms. They include bacteria and archaea. So under the heading of prokaryotic cells, the types of domains that we will find under prokaryotic cells would be bacteria, domain bacteria, and domain archaea. So these two domains contain organisms that have prokaryotic cells. They come in great many sizes and shapes. Most of the sizes range from about 0.2 to 1 micrometer to 2 through 8 micrometers. Remember, I've already taught you that the micro is like taking a meter and dividing it by 1 million. They also have some basic shapes. Cocci, which literally means berries. So that's where it gets its name from, because they're circular like berries. Then you have rod-shaped, or the bacilli, which literally means little staffs. Think of an individual holding a staff. You would think of it as in these shapes. And then you have the spiral. And because the formatting is off, this term that you see the pointer on, Vibrio, Spirillum, and the Spiraceae, those should actually be under the spiral. Okay? And the spiral, you have the Vibrio would be a curved rod shape. And then the Spirillum and the Spiraceae I've already discussed in lecture. In addition to the three basic shapes, there are some others. So we have the star-shaped cells that are of genus Stella, and this would be, you can see the cute, it's showing it in a pair here. Okay. Then we also have some squarish bacteria or rectangular, and those would be from, those could be halophiles. The rectangular flat cells are halophiles, which are archaea of genus Haloarcula. And we also have some triangular cells too. Most bacteria are what we call monomorphic, meaning that they maintain a single shape. So make sure that you add this, if it's not on there, add this to your study guide, monomorphic. And then you have some that are pleomorphic, which have many shapes, not just one. Prokaryotes not only come in different shapes, but in different arrangements. So these, they really, it seems almost as though they have some sort of cool personality. I mean, you can look at them and they look different based on which genus they're from. I just love these little critters. They have these different shapes, and so when the cocci divides, the cocci will reproduce and the cells can remain attached to one another. And if that is the case, you would have these here, which would be pairs. So diplo refers to pairs. Diplococci would be two cocci, or you'd have diplobacilli, which would be two rods that are stuck together. So they occur in pairs. Those that remain in pairs after dividing, again, those are called diplo. Those that divide and remain attached, as in a chain, like a sort of chain-like pattern, that would be strepto. So strepto would be referring to chains. And then we also have clusters, as in staphylococci. Those that divide in multiple planes and form like these grape-like clusters would be these staphylococci. So here we have the chain. We have, this would be a chain. We have a pair here. This would be the bacilli. So these are in chains. So streptobacilli, strepto refers to the chain. And in this situation, we have the clusters. You will not see the clustered rods because the rods tend to divide only along one particular plane right here. Because they don't divide on this plane that I'm drawing in blue, because they don't divide in such a way, you would not find clusters. This picture shows the typical prokaryotic cell. The components of the cells are grouped into three categories. You can have structures outside of the cell wall. You can have structures inside of the cell wall. And then you can also have the cell wall itself. And we are going to look at all of these structures. First, we're going to start off with the outside of the cell wall. So one of the structures that are outside of the cell wall would be the glycocalyx. Meaning, glyco means sugar. Hopefully you remember that. And then another term here is calyx, which means coat. So this is the sugar coat, essentially. It is secreted by the cell. If the secreted, and it's pretty sticky, and if the secreted substance is organized and firmly attached to the cell wall, then the glycocalyx is considered a capsule. The glycocalyx can also be in an unorganized and loose type of format, and in that case, it would be considered a slime layer. The glycocalyx helps the cells to attach to each other or to their environment, like a biofilm, how it can attach to their target environment. It's because of the glycocalyx. And when they're in the form of a capsule, this helps prevent phagocytosis. I kind of like to think of it as soap. You know how slippery soap is? That's what the capsule does for the microorganism. And if it invades an area where it is not, like for example our cells and our immune system, our hosts come to fight for us, if you have some cells that are getting ready to engulf this particular microorganism, that slipperiness will cause them to evade the phagocytosis many times. Some cells on the outside of the cell wall, once again, some cells have the flagella, which are long appendages that propel the bacteria. And these are made up of flagellin, which is a protein. Flagellin is a globular protein, really, and it arranges itself in a hollow cylinder to form a filament. So here is the filament. And attached to the filament is a slightly wider hook that you see me drawing in here. This is a hook and it's slightly wider, and it just consists of a different protein. The third portion of the flagellum is the basal body. The basal body I'll put in a brighter color here. I'll put it in green. This basal body is what anchors the flagella, and it anchors it to the cell wall and to the plasma membrane. You can see that this is the cell wall. Here we have the plasma membrane, and this basal body is what anchors it in there. Flagella come in different arrangements on the cell. So you have some bacteria. First of all, if a bacteria does not have flagella, so no flagella, we call that type of bacteria atrichous, which means without projections. But you can also have monotrichous, so of course that's just one projection. Or you can have lophotrichous, which is a tuft at one pole. And then we have amphitrichous, which is flagella at both poles. And then one of my favorites would be the peritrichous, where it's all over the cell. The flagella is all distributed all over the cell. I just wanted to point out on this particular diagram, which is one that you've already seen, is that gram-negative bacteria, they have a basal body that is larger. So their basal body is going to be much larger than a gram-positive because they have an outer membrane on the cell wall. Remember, this outer membrane is not going to be present in gram-positive bacteria. So because gram-negative bacteria have an outer membrane, their basal bodies are going to be much bigger. In gram-positive bacteria, the basal bodies are much shorter. So the flagella that we just talked about, those are used for motility. It allows them to move. We call it motility. When the bacteria moves in one particular direction, we call that a run. And then runs are interrupted by tumbles. The tumbles are caused by the reversal of flagellar rotation. If a bacteria has a lot of flagella, we call this swarming. The flagella are used to move the bacteria towards a stimulus, or it can also be used to move away from a stimulus, and this is called taxis. The ability to move is an advantage that enables the bacteria to move towards a favorable environment. If they're moving towards a chemical, that's called chemotaxis. If it's a light, then we call it phototaxis. The flagellar protein, which is called an H antigen, is useful in distinguishing among variations within a species. We call that serovars. There are at least 50 different H antigens just for E. coli. Spiraceae are different from spirals because they have a unique structure and motility. Treponema pallidum is a spiraceae, and it is the causative agent for syphilis. Spiraceae move by means of axial filaments, or something that we call endoflagella. The endoflagella, those are fibrils that arise at the end of the cell. They come from beneath an outer sheath and then they spiral around the cell. The rotation of the filaments produce a movement like a corkscrew. This is why we believe that this particular movement is very instrumental in the Treponema pallidum, which again is the causative agent for syphilis, to bore into the tissue of the individual. Many gram-negative bacteria contain hair-like appendages that are shorter and thinner than flagella, and these are called fimbriae. They are used for attachment. And then we also have pili, which are longer than the fimbriae and only have 1 or 2 per cell. They are involved in motility and DNA transfer. This is a process called conjugation. Conjugation, sex pili, the pilus of one bacterium called the F plus, and the F stands for fertility, will attach to another, which would more than likely be F minus, and the DNA from the F plus is transferred, giving the recipient cell a new function like antibiotic resistance or the ability to digest medium more efficiently. So again, the fimbriae, these allow attachment, and these allow the cells to attach to each other or to their surface, their target environment. This is why a biofilm is able to grow on surfaces because of the fimbriae. And the pili, those are used to transfer DNA from one cell to another through the F pilus. So far, we covered those structures that are in the category of outside of the cell wall. So why don't we take a closer look at the cell wall now? The cell wall is a really semi-rigid structure and is responsible for the shape of the cell. It surrounds the plasma membrane and it prevents the cell from bursting or lysis. If the water pressure in the cell is greater than that out of the cell can burst. And so the cell wall will prevent that. In bacteria, the cell wall is composed of a macromolecular network called peptidoglycan. Specifically, this is in bacteria. Other cells that have cell walls, they will be made up of another type of material, but for the bacteria, it is peptidoglycan. Peptidoglycan consists of a repeating disaccharide attached by polypeptides to form a lattice that surrounds and protects the cell. The disaccharide portion is made up of monosaccharides, and hopefully you can remember that from the biomolecule lecture. A disaccharide is 2 simple sugars put together. The monosaccharides in this case are what we call NAG, N-acetylglucosamine, and then we also have the NAM, which is N-acetylmuramic acid. And you can see just from this picture that the NAGs and the NAMs, so here we have a NAG, and then I have in green the NAM, and it's just repetitive, then another NAG, then another NAM, and you can see that these are held together by the polypeptides, which are the circles here. So that's what keeps these subunits of NAGs and NAM connected. Gram-positive cell walls have many layers of peptidoglycan forming a thick, rigid structure, and you can see it's illustrated here how it's several NAGs and NAMs. The gram-negative cell wall contains only a thin layer of peptidoglycan, as circled here. Gram-positive has teichoic acids, and the teichoic acids that would be an alcohol and phosphate and has a negative charge, which probably regulates the entry and the exit of cations. And the gram-positive has the walls act as antigenic specificity, and it makes it possible to identify certain gram-positive bacteria by certain tests. So the gram-negative has thin walls. It has no teichoic acids, and it has an outer membrane. Whereas the gram-positive cell wall is extremely thick and it has no outer membrane. In most of our last few lab meetings, we have done the gram stain. Electron microscopy indicates that the ability to resist decolorization or not is based on the different wall constructions of the gram-positive and the gram-negative cells. The gram-positive cells have a thick peptidoglycan layer, whereas the gram-negative cells have a thin peptidoglycan layer. The gram-positive cells have teichoic acids, whereas we see that the gram-negative cells do not. And the gram-negative cells have an outer membrane. The positive cells do not have that outer membrane. If the gram-positive cells are acid-fast, they would have mycolic acid. During the decolorization stage in the gram stain procedure, the alcohol acetone in the decolorizer stage, it extracts the lipid. So it makes the gram-negative cell walls more porous and incapable of retaining that crystal violet iodide complex more effectively. So when it washes away and we counterstain, it will remain pinkish, whereas the gram-positive cell walls will be able to retain that crystal violet within its cell wall because of how thick it is, and that will look more on the purple end. The gram-positive cell walls, again, they contain teichoic acids. They have lipoteichoic acids that link to the plasma membrane, and then they have wall teichoic acids which link to the peptidoglycan. And because of their charge, we believe that it may regulate the movement of cations, which are ions with a positive charge. The teichoic acids on the gram-positive cell wall has a negative charge because of the phosphate groups that are there. Also, we believe that it provides the walls antigenic specificity, making it possible to identify certain gram-positive bacteria by certain tests. The gram-negative outer membrane consists of lipopolysaccharides, lipoproteins, and phospholipids. The peptidoglycan wall is bonded to the lipoproteins in the outer membrane, and it forms what we call the periplasm between the outer membrane and the plasma membrane. The periplasm is just a gel-like fluid. The outer membrane has several functions. The negative charge evades phagocytosis and the action of complement, which is lysing cells and promoting phagocytosis. So it helps to prevent phagocytes from being able to engulf the cell. And the action of the complement, it protects against the action of the complement, which is lysing of the cell. Well, it will result in lysing of the cell or bursting. The LPS of the outer membrane has three components. One of the components is the lipid A. The lipid A is the lipid portion of the lipopolysaccharide. It functions as an endotoxin. What happens is that, of course, in gram-negative bacteria, when they die, they will release the lipid, and a fever can result, or the dilation of blood vessels, shock, blood clotting. All of these can be as a result of the lipid A being released. Then there's also the O-polysaccharide. The O-polysaccharide functions as an antigen, and it helps us to distinguish species. This outer membrane does not provide a barrier to all substances because nutrients have to pass through for the cell metabolism. Part of the permeability is due to the proteins in the membrane called porins. And they form channels that permit passage of molecules through the membrane. So here is a closer look at the gram-negative outer membrane, and you can see it consists of the lipopolysaccharides, the lipoprotein, and the phospholipids. The lipopolysaccharides contain that O-polysaccharide and the lipid A. The lipid A is the lipid portion, so the lipo portion, and then the O-polysaccharide would be the polysaccharide portion of the LPS, lipopolysaccharide. Also, here is the porin protein, so that substances can be regulated. The entry and the exit of them can be regulated in this outer membrane. And you can see that the outer membrane rests on top of the peptidoglycan wall, cell wall, and in between we have the fluid, or what we call the periplasm. So once again, the Gram-Stain mechanism takes advantage of the construction of the cell walls. In the gram-positive, the crystal violet iodine crystals will be able to form in the cell wall, which is much thicker than that of the cell wall in the gram-negative. The alcohol dissolves the outer membrane in the gram-negative, leaves holes in the peptidoglycan, and the crystal violet washes out. Whereas in the gram-positive, the crystal violet crystals, they are actually trapped within that peptidoglycan, that thick peptidoglycan layer. Among prokaryotes, certain types of cells have no walls or have very little wall material. Mycoplasmas lack cell walls. So those organisms that are of the genus Mycoplasma will not have a cell wall. By the way, mycoplasmas, those are some of the smallest known bacteria that can grow and reproduce outside of the living host cell. And because they're so small and have no cell wall, they pass through most bacterial filters, and they actually have been mistaken for viruses initially. Their plasma membranes are unique among bacteria in having lipids called sterols. And the sterols are believed to help protect them from lysis or rupture, which is great because they have no cell wall to protect them from that. So the sterols are believed to do that particular function. Archaea, those may lack cell walls, so they can be wall-less, or they have unusual walls composed of polysaccharides and proteins. But they're not peptidoglycan. They are not peptidoglycan. They do contain a substance similar to peptidoglycan called pseudomurine. The pseudomurine does not have the NAM subunits, the NAM. They don't have the NAM subunits like peptidoglycan. Archaea cannot be gram-stained, but appear gram-negative because they do not contain peptidoglycan. Chemicals that damage bacterial cell walls or interfere with their synthesis often do not harm the cells of the animal host. One way a cell can be damaged is by exposure to the digestive enzyme lysosome. This occurs naturally in eukaryotic cells, and we find it in perspiration, tears, mucus, and saliva. And the lysosomes essentially digest, or what they will do is, another term would simply be to cut. They cut up the cell wall like cutting a steel support of a bridge. So it cuts it, it cannot maintain its integrity, and then of course the cell can rupture. Particularly in gram-positive bacteria, the lysosomes will break down the cell wall components and makes them vulnerable to lysis. The cellular contents that remain surrounded by the plasma membrane may stay intact if lysis does not occur, and that wall-less cell is termed a protoplast. The protoplast can still carry on metabolism for the cell. When lysosome is applied to gram-negative cells, the wall is not destroyed usually like the gram-positive cells, but some of the outer membrane also remains, and in this case, the cellular contents, the plasma membrane, and the remaining outer wall layer are called a spheroplast. So that would be a wall-less gram-negative cell. For lysosome to have an effect on gram-negative cells, because remember, gram-negative, they do not have that huge peptidoglycan wall, that thick peptidoglycan wall. So for a lysosome to have an effect on them, the cells are first treated with EDTA, which weakens ionic bonds in the outer membrane, and it damages it. Some members of the genus Proteus, as well as other genera, can lose their cell walls and swell into irregularly shaped cells called L forms. And the L is named after the Lister Institute where they were discovered. They may form spontaneously or develop in response to penicillin or lysosomes. Now, when we move to the other category, which would be inside of the cell wall, first thing that we will encounter is the plasma membrane, which is also called the cytoplasmic membrane. It lies inside of the cell wall. It contains primarily phospholipids. Eukaryotic cells contain carbohydrates and sterols like cholesterol, but prokaryotic cells do not contain sterols, so their cell membrane is less rigid. The mycoplasma contains membrane, the sterols. The phospholipids are arranged in two layers that we call a bilayer. Now, hopefully you can remember that phospholipids are a type of lipid from the biomolecules lecture, and I'm going to just draw one right here. There's this phosphate head that has a charge, and then they have the fatty acid tails, which do not have a charge. I'll make them in a different color. If I had several of these and I just threw them into water, what you would see is them orient themselves just like what I'm drawing here. So you have the red phosphate heads and the blue tails. And what we will find is that if we just were somehow able to just shake these up and throw them into water, the tails will face away from the water. So they will face each other. And this is the inside, the inner portion of the phospholipid bilayer. And it does not like water, so we say that it is hydrophobic. Whereas those heads, they will face the water. They have a charge on them, these phosphate groups, and so we call this hydrophilic, water liking. There are also some protein molecules that are embedded within the membrane. Some are called peripheral proteins, as I'm circling here. Let me actually put my laser on it. These are peripheral proteins, and these are easily removed from the membrane by mild treatments, like really mild treatments, but their function would be for support, and they also work as enzymes. Integral proteins, which are some other proteins that you can find inside. Here's an integral protein. You'll see that it's bigger than the peripheral protein. The integral proteins that penetrate the entire membrane are called transmembrane proteins, and typically they will have some sort of channel that will have a pore, and this is to let substances in and out of the cell. The cell membrane, we call it a fluid mosaic model. The mosaic comes from the fact that there's several different aspects to it. There are proteins, there are sterols possibly, there are, of course, the phospholipids themselves. So, and it's fluid because the components move about freely inside of the membrane. They're not stuck together as a rigid, hard structure. So the membrane feels like olive oil. It has that type of consistency, and the proteins move around and function depending on their function. And you have the phospholipids; those rotate and move laterally as well. It's a self-sealing bilayer. If you were to take a pin somehow and stick it through the layer, it will open up for the pin to go through, and then once you take out the pin, it will seal itself back. It's self-sealing. Now, not stuck together. Remember, it moves about freely, but it will close. It will open around a needle, and it will close back when the needle is removed. Probably the most important function for the plasma membrane is the fact that it is a selective barrier. It selects which materials enter and which materials exit, and it's able to do that because of its makeup. Remember, the outside of the membrane is hydrophilic, the inside is hydrophobic. Large molecules also cannot pass through. So substances that dissolve easily in lipids like oxygen and carbon dioxide, those can pass through the membrane. The membrane also contains enzymes capable of catalyzing the reactions that break down nutrients to make ATP. Some bacteria contain pigments and enzymes as well that are involved in photosynthesis. You can find those in the plasma membrane called chromatophores or thylakoids. And these are some pigments and enzymes that are involved in photosynthesis. The plasma membrane is vital to the bacterial cell, so it is a target. Previously, we talked about the cell wall being a target, but it may or may not cause lysis. But if the plasma membrane is targeted, then it would be of detriment to. So we make antimicrobial agents to target the cell membrane. Detergents and disinfectants, those disrupt the membrane's phospholipids. Antibiotics, known as polymyxin, cause leakage of the cell's contents and eventually death. So remember that the cell membrane is responsible, one of its main functions would be to regulate what enters and what leaves the cell. So let's talk about movement across the membrane, because there are different types of movement across the membrane. First, we have simple diffusion. Simple diffusion is the movement of a solute from an area of high concentration to an area of low concentration. And you would see that most of the movement would be exactly that, from high to low concentration. So that's just simple diffusion. Simple diffusion is when, like carbon dioxide, oxygen, when the concentration is low in the cells but higher on the outside, if you need more oxygen, then the oxygen would simply diffuse across the membrane from an area of high to an area of low concentration. If carbon dioxide builds up in a cell, you have more CO2 here versus outside, low CO2, then the carbon dioxide will move from an area of high to low and your cell will get rid of it. That's just simple diffusion. Facilitative diffusion uses a transporter protein. It still is moving down the concentration gradient from high to low, but instead, if we were to draw, I'm going to just draw some membrane here very quickly, the phosphate heads and the tails. Now, if you have, here's the protein with that channel in it that we mentioned earlier, and then, of course, more of the membrane material around. In facilitative diffusion, you need to use a transporter protein where the material will be still going from an area of high to low concentration, but it cannot just pass through the membrane because of the membrane's makeup. So these can be things like sugars. If sugar is too big, then it will just move through these proteins, the transporter protein that is embedded in the membrane, but is still going from high to low concentration, down the concentration gradient. Another term is along the concentration gradient. This picture illustrates the use of a transporter protein to move materials along its concentration gradient. If we look at the little green circles, you can see that they're in high concentration outside of the cell, and on the inside, they're in low concentration. But they are too big to just pass through the membrane. That cannot happen. So instead, they use a transporter protein. The transporter protein will open, allow it in, and then it will close and push it into the cell. It's still moving, though, from high to low concentration. Now, we've been talking about cells rupturing and they can also lose water. So let's talk a little bit about the movement of water. Osmosis is the movement of water. And notice that specifically mention water. Okay, so osmosis is the movement of water, or we can say the diffusion of water, but it's still in the same way that you've seen simple diffusion or facilitated diffusion, and that would be still from high to a low concentration. Osmotic pressure is what will push the water through the membrane, because remember, water has a charge on it, and so does the inner portion of the membrane. So the water can either get through porins, aquaporins, which are proteins embedded inside of the membrane, or it can be forced through because of osmotic pressure. So this is just a nice little illustration of what would happen whenever when we're talking about osmosis. So when we're talking about osmosis, we're specifically referring to water, and we are specifically referring to the movement of the water through a selectively permeable membrane. So if we look at this setup, the very first setup that we have here, you can see that there is side A and side B. The little blue dots are water molecules. So if you were to really focus on this, you would notice that there is more water on this end. How do we know that? Because if you look at the purple, which are protein molecules, there are less of the purple. So on side A, we have 1, 2, 3, 4, 5, 6, 7, 8. We have 8 of the protein molecules shown here in our illustration. But on side B, we have 1, 2, 3, 4. So we have 4 of the protein molecules over here on side B. So if we have less protein on side B, then that means we have more water. And if we have more protein on side A, that means we have less water. So, by the way, just to make sure you understand, the water and the protein mixture, that would be considered a solution. So the entire thing is a solution. But what is being dissolved in the water is considered a solvent, a solute. So the protein is the solute. And what does the dissolving, in most cases it's water, that would be considered the solvent. So when we look at our little beaker here, we see that inside A, there is more solute, so less water. And then inside B, there is less solute, so that means there is more water. Water moves from an area of high to low concentration. So it will move from side B and go into side A, which is why when you look at the experimental setup, the water level has increased on side A because that's where the water movement took place. It moved into A. Okay, so now let's see what happens when the cells are placed into different solutions. We have some new terminology here to discuss. So let's look at the very first example. In an isotonic solution, isotonic means that there's on the inside and the outside of the cell, the solute concentration is the same. So solute concentration is equal. So I'm going to say inside equal outside. So that means that if the solute concentration is equal, that means that they have also, iso means same, remember that. So if the solute concentration is equal, then that means that the solvent or the water would be equal as well. So just for your understanding, I'm going to use numbers. If this had 60% solute on the outside of the cell and 60% on the inside of the cell, then I'm always going to do the water in blue. Then this means that it has 40% water on the outside of the cell and 40% on the inside because it'll always be a 100% solution. So because the water is equal on the outside and the inside of the cell, then the water will move equally in and out of the cell. There will not be any net movement. Water will still be moving, but it will move equally to the inside and outside. That's what happens when you place a cell in an isotonic solution. Our cells love isotonic solutions, animal cells do. Now let's see what happens with a hypotonic solution. So hypo is going to be referring to, hypo would definitely be referring to, hypo means less. You might have heard people say hypoglycemic, meaning that they have low blood sugar. Okay, so the term hypo is referring to less. So this means that the solute is referring to less solute. So I'm just going to write less solute right here. So now let's use numbers again, okay? On the outside of the cell, we have 60% solute. On the inside of the cell, let's make it 80% solute. So now let's do the math. If it's 60% solute on the outside, then that means it's 40% water and then 20% water. Okay, now remember, hypo means less solute in the solution. So if you compare the outer solution with what's going on with the cell, the outer has less solute. It has 60% versus the cell, which has 80% solute. So now where would water move? Water goes from an area of high to low concentration. So in this case, the water move into the cell. And this can cause the cell to burst. If the wall is weak, of course. If there's a nice strong cell wall around the cell, that will restrain absorption, and then the cell will be fine. But if that cell wall has been removed, then there will be very little integrity there, and the cell would explode. So I'm just going to illustrate it here. It just will explode. Of course, the picture shows it exploding way better than my little explosions that I've got going on here. Okay? Now, let's look at the hypertonic solution. If you place your cells in a hypertonic solution. So hyper, most of you have probably been around hyperactive kids, meaning that they're extremely active, more so than others. So hypertonic means that there's more solute in the solution. And we're referring to the solution that we put our cells into, or that the bacterial cells are being placed into. So now I'm going to jump back to our numbers. If the solution that the cell is in is hypertonic, that means it has more solute. So let's just flip this. Here's 80% solute. And then let's say on the inside of the cell, it is 60% solute. So our cells now are being surrounded by the 80% solution. So that means that our cells have been placed into a hypertonic solution. In this example, we've placed the cells. When I say our, I don't mean ours, even though this works for ours as well. But of course, I'm talking about the bacteria. So 80% solute means that there's more solute than what's in the cell, and this makes it hypertonic. Let's draw in the water. If it's 80% solute outside, then that means that it's 20% water. And if it's 60% solute inside of the cell, that means it's 40% water. So water moves from a high to a low concentration. It's 40% here, 20% here, so it's going to move outward. And that will cause the cell to pull away from the cell wall, and it would cause it to shrink. This is called plasmolysis, when the cytoplasm shrinks up. So as you can see, the cells will behave differently depending upon what type of concentration they are placed in. So far, we have looked at simple diffusion, facilitated diffusion, as well as osmosis, which is the diffusion of water. And we also looked at what would happen when the cells are placed in different solutions. That is, of course, we meaning isotonic, hypertonic, or hypotonic solutions, and the effect that it would have on the cell. But we're not done talking about transport yet. There's also another type of transport called active transport. In active transport, the substance that crosses is not altered by the transport across the membrane. But it does require a protein, and it will require the expenditure of ATP. Now, why? So far, we've mentioned that molecules move from an area of high concentration to low concentration. But in active transport, it's going to be moving from an area of low to high. That's not the normal way. Okay, this is not along the concentration gradient, but against the concentration gradient. So we call it active because it needs ATP to do that. It also uses a transporter protein, but the ATP molecule is what would open and close the transporter protein to move substances against or its concentration gradient or from low to high concentration. Now, there is a special type of active transport that occurs in prokaryotes. It's called group translocation. The substance that's entering the cell is chemically altered during transport. Once the substance is altered and inside of the cell, the plasma membrane is now impermeable to it because it wouldn't have receptors or anything or proteins to remove that particular type of substance because it's been altered. This allows them to take in substances even though they are in low concentrations outside of the cell. It will require energy supply, but it's not in the form of ATP. The energy is in the form of PEP, which is a high energy phosphate compound. Just like ATP is a high energy phosphate compound, so is PEP. And this is what would allow group translocation to transpire. Okay, moving along more about the anatomy of the cell, we're going to now look at the cytoplasm. The cytoplasm is inside of the plasma membrane. It is a substance that is made up of about 80% water. So there's quite a bit of water in here. It's mostly fluid. And then it's about 20% of proteins, enzymes, carbohydrates, lipids, ions. And the major substances are the nucleoid and the ribosomes. So the nucleoid is what you see here in purple. And then the ribosomes are the little dots that you can see, the little dots that are surrounded. Those are ribosomes. Now you will recall that in prokaryotic cells, they do not have a nucleus. No nucleus. A nucleus is what eukaryotic cells have. The DNA is surrounded by a membrane, and we call that a nucleus. But in the prokaryotic cells, they have something called a nucleoid region, where the DNA is just simply within the cell in a particular area. The DNA of the prokaryotic cells are long, continuous, double-stranded DNA that we call the bacterial chromosome. Both types of cells, both prokaryotic and eukaryotic, contain ribosomes. Ribosomes are the site of protein synthesis. Ribosomes are composed of two subunits, and by the way, on this pic, the ribosomes I pointed out earlier, but the ribosomes are these little dots that you see within the cytoplasm. The ribosomes consist of two subunits, each of which consists of protein and a type of RNA called ribosomal RNA. Prokaryotic ribosomes differ from eukaryotic ribosomes in the number of proteins and rRNA molecules they contain. They are also somewhat smaller and less dense than ribosomes of the eukaryotic cells. The prokaryotic ribosomes are called 70S. The eukaryotic ribosomes, by the way, are called 80S. The S refers to something called Svedberg units. And these Svedberg units essentially indicate how fast, if you were to centrifuge these, the rate of the settling of the sediments. The sedimentation rate is a function of the size, also a function of the weight and the shape of a particle. The subunits of the 70S ribosome are a small 30S subunit containing one molecule of rRNA, and then there's this 50S subunit, and that contains two molecules of ribosomal RNA or rRNA. We make antibiotics that will inhibit the protein synthesis on the prokaryotic ribosomes, and since they are targeted for prokaryotic ribosomes, they do not affect our cells' ribosomes, because remember, the prokaryotic cells' ribosomes are 70S, whereas our ribosomes are 80S. Within the cytoplasm of prokaryotic cells, There are several kinds of reserve deposits that we term inclusions. Some inclusions are common, whereas some are limited to a specific small number of species. So let's go through the different types of inclusions that we can run into in prokaryotic cells. We have metachromatic granules, and this is essentially a collective name given to large inclusions that sometimes stain red with certain blue dyes, such as methylene blue. They'll stain red. Volutin is a reserve of inorganic phosphates that can be used to make ATP. These are found in cells that are in phosphate-rich environments. Then we have polysaccharide granules. These consist of glycogen and starch, and you can see them when iodine is applied to the cell. And their function, once again, would be to act as an energy reserve. The lipid inclusions are found in Mycobacterium and Bacillus genera. These are lipid storage materials, so you can well imagine its function would be for energy reserves. And because the formatting is off on this slide, I'm just going to take a moment to sort of direct which type of inclusion goes with which function, because it's a little off, slightly off. Okay. Now we're going to move on to the eukaryotic cells. We've just finished the prokaryotic cells and their makeup, their anatomy, and the function of the different parts of their anatomy. And now we're going to move on to eukaryotic cells, which comes from the Greek word true nucleus. And eukaryotic cells really do contain a nucleus. The genetic material is enveloped by a membrane. Just as a recap, this is your typical prokaryotic cell, and you can see here that they have the nucleoid region, which is just DNA in a particular space. It is not surrounded by a membrane. You can see the ribosomes, the capsule that we talked about, cell wall. We also know that the plasma membrane is within the cell wall. And then you can see the prokaryotic flagella and the little pili, which help to transfer DNA from one cell to another. Hopefully you remember that eukaryotic organisms will include algae, protozoa, fungi, plants, and animals. The eukaryotic cell is typically larger and more complex than the prokaryotic cell. When you compare the structures of the slide before to the one that we have in front of us now, the slide before was a prokaryotic cell. If we compare it to the eukaryotic cell that we have in front of us now, there are some differences that become very apparent between the two. Now, in case some of you guys are looking at this thinking, wow, this is the weirdest cell I've ever seen, that's because it's a combination of two types of cells. I can draw a line right down the middle here. Okay, so this is actually I probably should just erase this and draw it downwards more of the, here we go. Right. So on this particular side, we have that of a photosynthetic cell with a cell wall. The rigid structure that you see here is the cell wall, and the green structures, I'll just write cell wall here, that's what gives it that rigid look. You'll notice on this side, there is no cell wall. And then we also have the chloroplast here. And you would notice that on this side, there are no chloroplast. If you were to look at it, really, this particular cell on, let's call it side A and side B. Side A is kind of like a typical plant cell with the cell wall. You see the vacuole here, the chloroplast, okay? Whereas on the side B, you have flagella, which is not in a plant cell, and no chloroplast, no cell wall. So again, this really cool picture is half plant and half animal. These cells don't exist. Half plant. It's just a composite type of sketch, and this is, the animal side. This is a diagram of the animal cell just on its own. It's not that composite half-half sketch anymore. And yes, you'll see that you have lysosome and you have the centrioles. These are not in most plant cells. They also lack a cell wall. They do have a flagella, which you do not find in plant cells. And you also do not have the chloroplast, which you find in plant cells. Here is your typical plant cell. And you would see that the shape is quite different. It's more rigid, and that is because of the cell wall. Very rigid. And it also has this huge vacuole, which you don't see in plant cells. In animal cells. They also have chloroplast. Please take note, though, that these plant cells definitely have mitochondria, and these mitochondria are for cellular respiration. So yes, plant cells perform cellular respiration as well. Just like the prokaryotic cells, the eukaryotic cells also have projections that are used for locomotion or for movement or for moving substances along the surface of the cell. The eukaryotic cells have flagella, and these are the projections that are long in relation to the size of the cell. And then there is also cilia, which are projections that are numerous and really short. Algae of the genus Euglena, they will use a flagellum for locomotion, whereas protozoa would use the cilia to move. Both the flagella and the cilia are anchored to the plasma membrane by a basal body, and both of them consist of microtubules. So the microtubules, they have about 9 pairs of microtubules arranged in a ring, plus another 2 microtubules in the center of the ring. So here are those that are arranged in pairs, and then central microtubules. Microtubules are long, hollow tubes made of protein called tubulin. So the tubulin is a protein. And again, they come in 9 pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 2. So that's the arrangement, the 2 in the center. We also call this a 9+2 array. Now you learned previously that the microtubules of the prokaryotic flagellum move in a rotation. They rotate. So they're going to be going in a circle-like manner. But the eukaryotic flagellum, those move in a wave-like manner to help propel the cell along. So to help keep foreign materials out of the lungs, we also have ciliated cells, and these move material along the surface of the cells in bronchial tubes. And you might have heard of a smoker's cough. Individuals who smoke, they have this dry hacking cough that they can't. The reason why they have that is because they have burnt the cilia off, and so now they do not have anything to efficiently move the phlegm upwards through their respiratory tract towards the throat and mouth. As far as the cell wall goes, you will see that the plants, algae, and fungi all have a cell wall. So this means that most eukaryotic cells have cell walls, but they are generally much simpler than those of prokaryotic cells. Many algae have cell walls consisting of the polysaccharide called cellulose, as do all plants. Plants have cellulose for their cell walls. Cell walls of some fungi also contain cellulose, but in most of the fungi, they contain a polysaccharide called chitin. So that would be for the fungus. They contain chitin. The plants and some of the algae contain cellulose. By the way, this chitin is what is the major material or the main structural component of the exoskeleton of crustaceans and insects. The cell walls of yeast contain glucan and mannan. In eukaryotes that lack a cell wall, the plasma membrane may be the outer covering. However, cells that have direct contact with the environment may have coatings outside the plasma membrane. In other eukaryotic cells, including animal cells, the plasma membrane is covered by a glycocalyx, which is a layer of material containing a lot of sticky carbohydrates. We've already talked about the glycocalyx when discussing the prokaryotic cells. Some of these carbohydrates that are on the or make up the glycocalyx are covalently bonded to proteins and lipids in the plasma membrane. These form what we call glycoproteins. The glycocalyx helps strengthen the cell surface. It also helps attach cells together, and it might also have something to do with cell-to-cell recognition. The eukaryotic cells, of course, also have a plasma membrane or a cytoplasmic membrane, which is the phospholipid bilayer as discussed earlier. It also includes the peripheral proteins, integral proteins, transmembrane proteins, sterols. So as you can see, the structure of this plasma membrane in the eukaryotic and the prokaryotic cells is very similar in function and in its basic structure. There are some differences in the types of proteins found in the membranes. Eukaryotic membranes also contain carbohydrates, which serve as an attachment site for bacteria. The plasma membrane of the eukaryotic cells are also selectively permeable, and substances can cross, again, both the prokaryotic and the eukaryotic plasma membrane by simple diffusion, facilitated diffusion, osmosis, or active transport. However, there was one type of transport that you learned about in prokaryotic cells. Hopefully you'd remember what it was. It still was an active transport, but the substance was modified as it was entering the cell so that it could not escape. The term was called group translocation. Prokaryotic cells, they have group translocation. However, eukaryotic cells do not have group translocation. However, eukaryotic cells do have something that is unique, where it can move substances through the mechanism called endocytosis. Endocytosis comes in three types. There's phagocytosis and pinocytosis, and there's also receptor-mediated endocytosis, which is not shown on this picture. So in phagocytosis, which is used by the white blood cells to destroy bacteria and foreign substances, you would have cellular projections that are called pseudopods. So this should have a D at the end. You have these structures called pseudopods that engulf particles and bring them into the cell. In this particular case, on this diagram, you can see that this particle, the green particle, is being brought into the cell. This would be phagocytosis. Pinocytosis, the plasma membrane folds inward and it brings in fluid into the cell, along with whatever substances are dissolved. So pinocytosis is more like cell drinking, whereas phagocytosis is more like cell eating. Now, when we look at the cytoplasm of eukaryotic cells, that will include, when you hear the term cytoplasm, that includes the substances inside the plasma membrane and outside the nucleus. So this cytoplasm pretty much includes the liquid and everything else that's inside the plasma membrane and outside of the nucleus, but still within the cell. The term cytosol refers to the fluid portion of the cytoplasm. So if you were to think of a bowl of Jell-O, the bowl of Jell-O with fruit in it would be the cytoplasm, but just the Jell-O alone would be the cytosol. I would say that the biggest difference between, or maybe one of major difference between the eukaryotic cytoplasm and the prokaryotic cytoplasm is that the eukaryotic cytoplasm has these internal structures that look like small rods that are called microfilaments or intermediate filaments, and they also have microtubules, which are little cylinders. So that cytoskeleton, it provides support and shape, and it assists in transporting substances through the cell. Kind of acts like a railroad, and it helps get substances from one end of cell to the other. The movement of eukaryotic cytoplasm from one part of the cell to another also helps distribute nutrients and move the cell over a surface. and this is called cytoplasmic streaming. Now, the eukaryotic cell consists of many organelles. Organelles are structures that have specific shapes and they have specialized functions. They do things for the cell. And a lot of times, if something is wrong with just one organelle in the cell, it can cause some serious problems. It can even cause death. So these organelles include the nucleus, which contains chromosomes. Then there's the endoplasmic reticulum, which is this full-on transport network. You have the smooth ER and the rough ER. The rough ER is rough because it has ribosomes on it. Then you have the Golgi apparatus or the Golgi complex, and that is a membrane formation. It helps out with membrane formation, and it also secretes whatever was made by the cell. There's the lysosomes, and these contain digestive enzymes. As a matter of fact, if you've ever heard of Tay-Sachs disease, this is a genetic disorder, and what happens is the lysosomes, they lack the enzyme that will break down lipids. So think of it like a garbage bag that you keep shoving full of garbage. Eventually, it's going to burst. Well, this is what happens with the lysosomes. They keep getting shoved full of this lipid in these young children who have Tay-Sachs disease, and then eventually the lysosomes burst. But remember, there are other enzymes in the lysosomes, and if that's unleashed on the cell is destroyed. And this is why these young kids typically won't make it past the age of 5 or 7-ish. Also, there are vacuoles, and the vacuoles bring food into the cells. They also provide support. And in the case of like a paramecia, they have contractile vacuoles that will pump water outside of the cell. There's also the organelle called mitochondria, and this is responsible for cellular respiration, and chloroplast responsible for photosynthesis. In addition, there's another little sac that you can find, like the lysosomes, that are called peroxisomes. And these oxidized fatty acids, they also destroy hydrogen peroxide, which is toxic to the cell. There are some structures within the eukaryotic cell that are not bound by a membrane. One is the ribosome, which is responsible for protein synthesis. Then there's also the centrosomes, which are located near the nucleus, and these consist of protein fibers and centrioles. The centrioles have what we call a 9-0 arrangement or a 9-0 array. Earlier, you learned about the flagella having the 9-2 arrangement. Well, the centrioles have the 9-0 arrangement, and the 9 refers to the 9 clusters of microtubules, and the 0 is the absence of microtubules in the center. The centrioles are responsible for cell division during mitosis and meiosis. Now, at this point, I would strongly urge you to go to your textbooks or look at the lecture notes that you have in front of you so that you can see what each of the structures look like that are within the eukaryotic cell. I essentially gave you a straightforward function of each of them, but again, if you want to know greater detail, you can look at your original PowerPoint presentation that was posted, and you can see the different organelles and internal structures, and then you can go to your books and read up even more on their functions. Now, I'm going to talk a little bit more about these ribosomes because they sort of indicate to us the evolution of eukaryotes. So remember that the eukaryotic cells, that they have what we call the 80S ribosomes. I discussed these 80S, the Svedberg units, earlier when talking about prokaryotic cells. The eukaryotic ribosomes are membrane bound, and they're attached to the endoplasmic reticulum. They can be. Whenever they are attached to the endoplasmic reticulum, we call that the rough ER. So you'll see rough sometimes in front of the endoplasmic reticulum, and that's because there are ribosomes attached to it. And then there are some ribosomes that float freely throughout the cytoplasm. Now, when we look inside of the cell, though, we find that chloroplast and mitochondria, these two organelles, they're very unique in that they have their own set of ribosomes, and they are 70S. Remember, 70S ribosomes are the types of ribosomes that prokaryotes have. So within the eukaryotic cell, the chloroplast and the mitochondria have 70S, which is very similar to the prokaryotic ribosomes. So this now leads us to the endosymbiotic theory. It is believed that the eukaryotic cell originated as a community of prokaryotes. So the eukaryotic cell more than likely came from prokaryotes. And we believe that this happened more than 2 billion years ago. So it happened in two phases. First, there was the membrane enfolding theory, meaning that you have the cell, a prokaryotic cell, and then the membrane material folded inwards and sort of created, here we go, just kind of made what we call invaginations. And there you have it. And we believe that what would have happened is when that membrane enfolded, it would have wrapped itself. Let's say this was the nucleoid region, so some DNA there. Now, this membrane enfolding would have wrapped itself around this nucleoid region. And now it would not be a nucleoid region, it would be a nucleus because it is genetic material that is bound by membrane. So that's a true nucleus. So the first part of this theory is the enfolding of the plasma membrane. So when we talk in biology about symbiosis or the symbiotic relationships, that's a close association between two or more species. So endosymbiosis would be, endo would be inside, symbiosis would be living within another, so, or living together. So endosymbiosis would be one species living within another. The mitochondria and the chloroplast are believed to have once been prokaryotes. So after there was the invaginations or the enfolding of the membranes, then it is believed that one huge prokaryote probably engulfed a smaller prokaryote. And here, let's just move it on in. So now you have it here, and I'm doing this green to illustrate that it was some sort of prokaryote that could photosynthesize. And then, of course, that would have gotten more membrane material around it because of the invaginations. And then you would see that now that prokaryote that used to be independent, so to speak, was now engulfed, membrane material wrapped around it, and now it is a chloroplast within a eukaryotic cell. And the same would also go for the mitochondria. Here you have the mitochondria, and it might have been engulfed, and when it did, here it is, and then it would have gotten some of that material, plasma membrane around it, and now you have a organelle that we now call the mitochondria, but it was at one point an independent prokaryote. So these smaller prokaryotes were engulfed by a bigger one, and when we look at the molecular systematics, the genetics of it, we can see that chloroplast and mitochondria kind of rock to a beat of their own, which closely resembles that of prokaryotic cells. Also, the flagella, we believe, arose from some sort of Spiraceae bacteria, and again, it probably was, hanging out and then got engulfed and got embedded in the plasma membrane, and there you have your flagella. This is actually supposed to be just one flagella. We're not like, eukaryotic cells are not like prokaryotes where they can have a whole tuft of them. So let's do that over. There we go. So here is a diagram illustrating what happened. First, we have the invaginations that would have taken place, or the enfolding of the membrane. And so when this membrane folded around what was once the nucleoid region, it created what we would call some sort of rudimentary nucleus. So this would be the nucleus that was created. Then the smaller prokaryotes that you see here, this one is a prokaryote, a heterotrophic prokaryote. Here we have a photosynthetic prokaryote. These were engulfed by a bigger prokaryote, this ancestral prokaryote, and then eventually they lost their independence and they are now considered organelles within the prokaryotic cell. So once again, this is called the endosymbiotic theory, and we have molecular evidence to help support it. So this pretty much wraps up I know I left a few slides out, but I just wanted to shorten this as much as I could, just hit on the main topics within this chapter 4. And this wraps it up. If you have any questions, any comments, of course, I would love to hear them. I would love to be able to address anything that still is of concern to you. But in the meantime, happy studying.